Hello, and here we are. So it's going to be the second part of the first lecture on chapter 4 about cell physiology. So I've already introduced things like anabolic and catabolic reactions and how we can make anabolic reactions happen by using ATP, or coupling a reaction that needs energy with one that releases energy. So let's continue. Does this look familiar? Yes, ATP, we talked about. Went through the numbers, but that's not important there. So these all these reactions that take place, and I show you one here that we will actually get to. And yeah, depending on your background, you may have had a teacher that was more into the biochemistry that had you learn all these parts, all these different intermediate chemicals, and each one has an enzyme that catalyzes that reaction. So you can memorize that, and in some classes you do. We're not going to do that. I'll teach you what comes in, what comes out, and how much energy comes in and out. So not all the details, but this is looking at a cycle that goes around that keeps producing the same beginning, and then it goes around and it spits out molecules along the way. So things go into this cycle and they go around and around and around like that. So, and what controls that? Well, I told you enzymes at each step do for sure. So that hump, the hump you have to get over. So this reaction is spontaneous, meaning that A is going to go to B because, and it's going to give off energy because you're going downhill. But you've got that activation energy you've got to overcome. And mainly, if we're talking about two molecules that they're going to bind together to make one, in a solution, the molecules have to be close to each other for that to happen. They've got to be in the right alignment, and then they'll start sharing those electrons or whatever they're going to do. So how can we bring them together more often? Well, one thing is turn up the heat. And so the more heat there is, the more the molecules are moving, the more likely they're going to collide with another. And if they do in sufficient energy, that reaction will take place. So reactions take place quicker at higher heat to a point. But the other way, without introducing heat, is using enzymes. This is a cool way to do it. Oh, sorry, that was bad. But what they're going to do, enzymes are like matchmakers, and they will take the two molecules, and they'll put them close together, and they'll kind of force them to come together. And they do this thousands of times per second. And they encourage the reaction to take place. They lower that activation energy so it takes place much more easily. And so in our bodies, although we have reactions that would take place, it'd be far too slow without enzymes, so we couldn't survive. So we have all these enzymes, which are proteins coded by our genes, that are being used in all these chemical reactions to chaperone them along, to make them happen more rapidly. So showing you here, this big blobby thing is the enzyme. It's a protein, and it's got two active sites that are just perfectly shaped. So in this case, a sugar fits perfectly in one, and the other one, this ATP molecule, fits in like that. And as soon as they both fit in those little slots, the whole protein changes shape, and it brings them together right next to each other in just the perfect alignment where that phosphate goes over to the sugar. And as soon as that happens, the protein changes again, and those things get popped out. And then it's free to do it again and again and again. So that's how enzymes work. They got to be just the right shape. And then in that case, we're putting two things together. But enzymes can break things apart too. Like let's say a certain sugar fits into an enzyme, and then that protein, the enzyme changes shape, and it stretches that bond, and it causes them to go apart. So enzymes can put things together or break things apart. And they're not used up. They're used over and over again, but they're just like a matchmaker. They put the molecule in the right alignment so that the reaction happens easier. It would happen without the enzyme, but just so slowly. You'd have to wait for the molecules to hit in just the right configuration. This puts them together just perfectly. That's what enzymes do. And look at graphically, it's going to lower the activation energy. That's what enzymes do. You can look at this animation from the PowerPoint. You can click on it. It just looks nice. You don't need to see it right now. Yeah, and this is showing. So the substrate is going to be whatever you're working on. Let's say we're breaking down some kind of sugar, and the enzyme has an active site that it fits in, and then the products come out, and then it's ready to go again. You recycle this enzyme, use it over and over and over again. Yeah. Sometimes there's control on the other end. There's another site that can turn on or off an enzyme if a reaction is happening too quickly or you want to speed up, slow it down. So there's some subtleties there too, but you can't get into everything. Beautiful. And here's an enzyme putting two things together. So we call it the substrate. The products are what you're forming, and the enzyme is left unchanged. So the timing of it is that you have the substrate is going to attach to the enzyme. Then you have this complex that for a moment you have the substrate and the enzyme bound together, and then it's going to either bring them together or break it apart, and it's going to release them, and you have a product. And so this goes round and round and round. So the shape of the protein is so important, and that's why most genetic diseases are because you have an enzyme that's not properly shaped. Remember, an enzyme is a protein. Proteins are made from the genes, right? The genes code for proteins. So if you got a wrong letter, your protein's not going to be the right shape, and it's not going to work. So the shape has to be just right. And enzymes are specific. Usually they just fit the one kind of molecule. And there's a whole pathway, and you can see here from going from the beginning to the end, this is more realistic. You have many intermediate chemicals on the way, and each one has an enzyme. And so this is where diseases can come in, where if one enzyme is screwed up, damn it, you can't get to where you're trying to get to. And you end up backing up with some kind of product that's not the final product. And so it's an issue. Yeah. Enzymes usually have the ACE, so sucrase breaks down sucrose, sugar. Lactase breaks down lactose, sugar. A lipase breaks down lipids. Proteases break down proteins. So anything ACE is it's going to be an enzyme for that. And how is this controlled? Usually, you want the reaction to happen at a certain rate. You don't want it to happen too fast or too slow, and you have control there. And usually there's a negative feedback, where if you're making too much of the product itself usually inhibits the early enzyme. I say early because if it just went here, you'd end up going all the way there, and you end up with a backup of some product. So usually it's best to go down back to the beginning to limit this pathway. A couple of things also, you can have cofactors and coenzymes. Cofactors are non-proteins. So these are often small molecules that are involved in controlling enzymes. They attach, you can see, to not the main sites, not the active site, but another sites, allosteric, a different site. Then it's going to change. Imagine if it attaches somewhere else on the enzyme, changes the shape of the enzyme that can no longer work, right? You remove it, all of a sudden it goes back to that original shape and it can work. So it's kind of a control center. And coenzymes are organic factors. You need in small amounts, vitamins or things like that are gonna work. So cofactors, coenzymes are small controlling molecules that control the enzymes. Look at this thing. This is catalase. Oh my God, I'll talk about that when I talk about a particular cell organelle that breaks down hydrogen peroxide. But here's hydrogen peroxide. Look at the size of it. So the protein, this enzyme is huge, right? But that hydrogen peroxide is going to fit into this active site, and it's going to break it apart and release oxygen and water. So heats, of course, the more the heat, the faster the reaction will take place. But indeed, yeah, radiation, electricity, chemicals, pH. So for your enzymes to work, they have to have the proper conditions. And that's why you have homeostasis in your cells, where the pH is kept at a certain level. Your body temperature is where the enzymes work optimally. So if you look at the activity of an enzyme, and you can see that when it's cold, it doesn't work so well. But guess what? That's probably going to be, I'll even use Celsius. It's going to be body temperature. It works the best. And then if you get too hot, then the enzymes start denaturing. Remember that? The proteins break apart and they're never going to come back. So enzymes have an optimal temperature, an optimal pH, everything. And that's where we try to keep our bodies at. All right, a little big picture. Talk a little bit of physics, talk a little thermodynamics here. But we're talking about energy, right? It's to power your thoughts, your athletic endeavors, everything. And energy is the ability to do work. I'm thinking, I love physics actually. Chemistry, yeah, but physics, I really like that. And we look at energy, potential energy is the energy in a bomb, an energy in a can of gasoline, the energy in a boulder at the top of a hill, the energy in that water that's up in a water tower, that gravity brings it down. So that's potential energy. And you know, we use it, the chemical energy stored in fats and carbs. That's potential energy, right? And kinetic energy is with movement. A kinetic sculpture is one that moves in the wind kind of thing, yeah. So looking at throwing a baseball, it's got kinetic energy when you're throwing it up in the air, when it's moving the fastest. And then it goes up, the kinetic energy slows as it gets slower. And when it's at the peak, it has the most potential energy because then gravity can work on it. And then the kinetic energy starts growing as your potential energy gets less as it comes closer to Earth. Just some examples. So why is this important? Well, we use the chemical energy held in bonds in our food to power all of our chemical reactions and our muscles and our thoughts and everything. Yes, laws of thermodynamics, love it. The first law, energy is not created or destroyed, but it can be transformed from one form to another. Right, you guys got that? So you can't make energy out of nothing. You can't destroy energy. It can just be changed to different forms. So you light that bonfire, you're taking the chemical energy in the wood and turning it into to heat and light. You're transforming it. Cool. And the second law is that this idea that everything goes towards greater disorder. It doesn't naturally become more ordered. Your room becomes more messy normally. It doesn't all of a sudden clean itself up and organize itself, right? It takes energy to do that. So it's entropy. Entropy is the degree of disorder, and it always increases in a system, in the universe, naturally. So indeed, your room always goes towards more disorder. Rarely do all your pencils become ordered by size and put together. They normally end up all over the place. So yeah, so that's the deal. Here's highly ordered, more disordered. And that's what the second law of thermodynamics is that things go towards more disorder in the universe. I mean, a big picture, the Big Bang, things are becoming more disordered in the universe over time. So people often look at life, and they even talk about evolution as being impossible because it goes against the second law of thermodynamics. Well, that's crap. They're not looking at it correctly. And so you think to yourself, on this planet Earth, how do we make such complex organisms like the human beings and all life, plants, everything? It's more ordered. So is Earth violating thermodynamics by becoming more complex and ordered? Well, if you just looked at the Earth by itself, yes. But in terms of the big picture, the whole system, we are increasing entropy in the whole system because the whole system is heating up. So locally, we're getting more complexity when we make a mouse or a person. But overall, in making that, we have created more disorder in the rest of the universe. Am I getting too big picture for you? Yeah. So life goes against the second law of thermodynamics, seemingly, until you look at it. Overall, you're increasing entropy in the universe by creating this complex person. Yeah, I think I have, yeah. Yeah, this kind of shows it. So you can see here that, sure, you've built up complex molecules. You've created less entropy. You've gone against it. But in doing that, you've given off so much heat, you've heated up the environment that you have, in fact, increased entropy when you look at the whole system. So we build up complexity that goes against entropy, but we've actually increased the disorder of the whole universe around us by doing it. So we haven't violated that law. All right, and then here's ATP. you should know this molecule is it's critically important. It's adenosine triphosphate, so it's an adenine molecule. It has a ribose sugar, and it's got 3 phosphates like this. And this is our energy. This is the molecule that powers our reactions, that makes your muscles go, that runs the pumps, that keeps the nerve polarity, the sodium potassium pumps. Getting too. It's gonna run everything as ATP, and it's like a rechargeable battery. It's that energy in that 3rd phosphate. When you break that bond, it gives off energy and it powers reactions. And you're left with a lower energy ADP, adenosine diphosphate, with 2 phosphates. And that's low energy, but you can use energy to put another phosphate on to recharge it back to the ATP. And this is what we do. It's a rechargeable battery. We make ATP, we use the energy, we get ADP, we recharge it ATP, and that is used constantly. And we burn sugar, as long as it has oxygen. Even without oxygen, we burn sugar, and we are going to be able to recharge that ATP. Yeah. So we change energy from one form to another. We eat a cheeseburger, we get fats, we get carbs. We're going to change that chemical energy into another form. We're going to take the energy in those bonds and put it into the bonds of ATP, and then we're going to use that to make things happen. And whenever you change energy from one form to another, you lose a little in heat. So it's not that efficient. I don't know if I need to get there, but heat is given off in these reactions. And so it's never 100%. You can't transfer one energy to another without losing some heat. Yep. So we burn glucose, we oxidize it, and eventually all the carbons in sugar become carbon dioxide, which is a waste. And then plants take that and make sugar again. Thus, it goes around. All right, so basically the cellular respiration, which I will talk about in lecture. I want to get to you guys in person, be able to explain it, answer any questions. Again, you guys should know it. You can review it. And if you're reviewing it, you look in the book, don't get caught up in the details of memorizing all the intermediate products and all the enzymes. I care about big picture. What molecules do you start with, do you end with? How much energy do you get in each of these 3 steps? Okay, if you're looking ahead for that. And I'll tell you that in person too. But basically, we take sugars, and because we're not a plant, we can't make sugar. So we steal it by eating plants or eating animals that eat plants. We steal it, and then we can burn sugar to get energy, to get ATP. Turns out if we have oxygen, we can get out, I'll give you a preview. Just burning sugar without oxygen, we can get like 2 ATP. If we have oxygen, we can get like 36. So in a pinch, we can burn sugar without oxygen, get a little bit of energy, but not enough so that we can survive like that. You know, if you're doing a sprint muscles will burn sugar, but you got to recover. You can get cramps, things like that. So oxygen allows us to really burn that sugar and get all the energy out. So what goes in? We need sugars and oxygen. And honestly, you can, if you guys eat a carb-free diet, you can live on fats and proteins. We can also break down we can change that. But just for simplicity, we got glucose in the presence of oxygen, allows us to burn it, get all that energy, and we get carbon dioxide and water, and that energy that we store as ATP. Yeah, so just 2 more slides here. This shows kind of graphically how we get ADP here, and then using energy, we're going to see, harnessing the energy when we burn some sugar, we can put that phosphate on. It takes energy, but we can put it on. And then we end up with this high energy ATP. And that is at the top of this curve, this energy hill. And we can use that if we break that phosphate off, it gives off energy, brings it down again, but that energy we can use for everything. So this is the key. Then this ADP gets recharged. How nice is that? So it's a rechargeable battery, this molecule. With the 3 phosphates, it's high energy. We take the phosphate off and we use that energy, and then it's recharged. All right, so I'll see you guys in person, and we'll talk about these 3 steps of cellular respiration. Now, respiration sounds like breathing, but here it's a different form. We're talking about cellular respiration is how we break down molecules like sugar to get ATP, to get energy. And you guys can study ahead of time for the lecture and know it has 3 parts. And we'll go through each of these in person. All right, wonderful. Hope you guys are having a good day. I don't know when you're watching this. Good night, whenever you happen to be watching this. But I will continue this little lecture in person.